Okay, so every single tissue and cell is going to have multiple arteries and veins that are either going to be supplying if it's an artery or draining blood if it's a vein from that area. So there's always going to be these alternate routes, okay, or like detours. Now, these detours in terms of vessels are called anastomoses or anastomosis if it's singular. Okay, so we're looking at a net of vessels. So, really, the only one that we're going to talk about uh, in this class is the circle of Willis, and that's in the brain, but we'll get to it in another video. But just pointing out that these anastomoses, these are detours, these are alternate routes. And it's really just like a protective measure because sometimes arteries get blocked, sometimes veins collapse, okay? And there always needs to be some way for blood to reach the tissue or the cells or the organs. Now, when we are reaching the tissue and the cells and the organs, it's going to be through the capillaries, right? So capillaries are organized into capillary beds, and capillary beds is kind of what you're seeing here. A little histo image of that, right? So I don't know the magnification, but super blown up, right? But up in the capillary beds, there's up to about like 100 capillaries per bed, right? And then they have all of these different sphincters surrounding the capillary bed that can actually change the direction of blood flow depending on the need of the tissue the capillary bed is supplying. Okay, so think about it. So every single cell in the body, almost every single cell in the body, is going to have some connection with a capillary, and you know, there in turn a capillary bed. Okay, but sometimes the needs of the cells are going to be different at varying points of the day, and you know, depending on body usage, right? So depending on the need of the cell and the need of the tissue, sometimes blood doesn't need to be in full force or something like that. So these sphincters can redirect blood to kind of maybe bypass that area. Okay, so it's pretty cool. It's like the most advanced, construction crew, with their flags and whatnot, a flagging crew, right? Directing the cars essentially. I always think of the vessels like a highway system almost. But anyways, when we're looking at the capillary bed, we can see that we have these arteries and that leads into atrioles, right? Or arterioles, excuse me. And that leads into something called a met arteriole. And that is going to be the main source, main channel of blood into the capillary bed. Okay. And then there's something called a thoroughfare channel, which is essentially, it looks like a capillary, but it's a channel that's providing a different route for blood from the atrial to the venule, and it can actually bypass the capillary. Okay. So, again, directing blood flow through the help of the thoroughfare channel and the sphincters, depending on the need of the tissues, and also helping regulate blood pressure. So that's pretty cool. All right. So just kind of surmising what I just said, right? So capillaries are into capillary beds. Cool. They're going to be supplied by a single met atriole. Great. We have that thoroughfare channel that can, bypass the capillary bed and, direct blood right to the venule. Okay. We have the sphincters. You don't have to, identify really anything like this. I wouldn't ask you, where's the thoroughfare channel? Where are the precapillary sphincter? I'm not going to ask you any of that. But just kind of be aware of it, right? So when the sphincters are open, blood can, we can exchange with the gas exchange with the tissues. When they're closed, it's going to bypass the capillaries. And again, depending on the need of the tissue. Okay. So really, when you think about it, 3/4 of the body's capillaries are going to be shut down at a given time because of, the needs and these sphincters being closed and whatnot. Cool. So there are 3 types of capillaries, and they're in different locations. Okay. So you need to know their location, and you need to know the structural differences between these 3 types. Okay. So the first one here, the name kind of got cut off. I'll try to fix that. That this is a continuous capillary. Okay. Then we have a fenestrated capillary. And then we have a sinusoidal sinusoidal capillary. Okay. Again, let's talk about their differences structurally and then where they're located in the body. Okay. So continuous capillaries, it kind of gives away right there in the name what we're talking about, what we're looking at. We have a continuous basement membrane, which is this sheer portion here and kind of being shown in blue. That's a basement membrane. So this continuous basement membrane, so the endothelial cells are all going to be continuous. There's no gaps really, except for something called intercellular gaps. So you can see the cells and you can see the lines separating each cell, right? There are these gaps called intercellular clefts. Okay. And that's where things can actually diffuse through. Okay. But by all intents and purposes, these are continuous capillaries. Now, these guys are going to be located in or around the central nervous system, the lungs, and muscle tissue, and also the skin. Okay. Fenestrated capillaries. So, fenestration, where it gets its name, is referring to pores. All right. And that's what we're seeing here. We have these very large pores called fenestrations. Well, they're not that large, but all right. So, we have these small holes basically. And these guys are going to be located in the kidneys, the villi of small intestines. Oh, the choroid plexus, right, where cerebral spinal fluid was formed in the brain, in the ventricles. Also, most of the endocrine glands as well will have fenestrated capillaries. Okay. And then the sinusoids, capillaries, these guys are going to have really unusually large fenestrations, right? So I don't want to say that these are large because these are large down here. So we actually have, again, those intracellular gaps. We can see they kind of follow the lines, but they are massive gaps. And then we have the fenestrations in the actual cell, and those are also very large. So a combination of these large holes essentially, plays into the sinusoidal capillary. All right. So this is going to be allowing more things to diffuse more easily, right? Or even enter easily as well. So when you have like new blood cells being formed and then they need to enter the bloodstream, right? You would want sinusoidal capillaries in that location so that they can easily enter, right? I mean, picture a red blood cell trying to get through a fenestration over here, right? It wouldn't work. So we would need this type of capillary in the location where red blood cells are being formed. So that's going to be red bone marrow. So that's one location. But they're also going to be around the spleen, the anterior pituitary gland, parathyroid gland, adrenal glands. Yeah. Okay. So lots of glands here. Okay. So we do need to know, just kind of, either being able to identify them. Maybe I talk about some of their, structural differences or talk about the location. You need to be able to, tell me what type of capillary I'm describing or something like that. Awesome. So veins. Let's just kind of recap on veins a little bit because when we were going over the structure of veins versus arteries, veins were a little bit weirder. Okay. So veins again are formed when venules converge. Cool. They have the three tunics, right? But the media is pretty thin because it's lacking all of that smooth muscle. It's still there, but it's not as thick. And then we do have a really large lumen. Cool thing about veins, they are considered a blood reservoir. Okay, and you could either call it a blood reservoir or uh, capacitant capacitance vessels. Okay, but blood reservoir is just kind of easier. Excuse me. So basically, in veins, blood can be diverted really quickly, right? Um, so about 65% of the blood supply at any given time is found in the veins. Okay, but the other weird thing about veins was that they had valves. Okay, so when we're thinking about arteries and we're thinking about how blood moves through the arteries, right? Arteries are directly off the heart, they're moving blood away from the heart. Blood is being pumped because of the force from the heart. So that's why arteries have such a higher blood pressure, because it's this massive force from the heart propelling it throughout the systemic circuit. Okay, but by the time blood gets back to the veins, right, it goes through the capillary beds to the venules and then goes up into the veins, the pressure is pretty much lost at that point. I mean, there's still some pressure, but it's pretty much lost. So veins are going to veins are going to depend on something called the muscle pump. So looking at skeletal muscle, we can see here we have muscle contracting, right, which is actually going to push venous blood up, okay, towards the heart. All right. But let's say the muscle relaxes before the blood can be expelled all the way through, right? It's going to fall back down, but it doesn't, keep going. It doesn't pool anywhere because there are these valves, right? So once the muscle relaxes, the blood kind of gets caught, and then maybe the muscle contracts again, pushing the valve will open and the blood can be expelled, right? But these valves help prevent backflow and blood pooling in the body. Okay. And so sometimes valves do fail in the veins, and then you can have, a few things happen, but like varicose veins or something like that can occur because the valve fails and the vein can, collapse and whatnot, and some varicose veins can form. But anyways, that's a whole other thing. This is just kind of showing a histology of the valve. So you can see it is a little bit different than the valves we looked at in the heart. There's no choroid tendinoids or anything. There's no muscle that anything's touching, like papillary muscle or anything. It's just we have these valve leaflets, okay, on the opposite side of where blood would be flowing. Okay, but it's pretty cool that you can actually see that on the actual histology of it all, right? And this is the really blown up version here, so you can see those leaflets. Okay, cool.